you, Aaron. So it's my pleasure today to introduce Oscar Serrano. So Oscar is a fellow Catalan, like myself, and he started his PhD just as I was finishing in the same research institute, which is called Centre d'Estudis Avançats de Blanes, just near Barcelona. The PhD, like the actual thesis, goes through the University of Barcelona, but the research goes through this institute, which is the equivalent of a CSIRO type institute. But I've heard lots about Oscar, and I have met him through the years. So I was reading a little bit about him this morning on the internet, and I came across this kind of quote about Oscar Serrano being a fortune teller. But instead of reading palms, he reads the history of human and climate impacts using seagrasses, which is quite a beautiful way of putting the kind of research that he does. And I'm a big fan of how he combines natural history, paleoecology, and then just kind of, everyday ecology. So he finished his PhD in Blanes and the University of Barcelona in 2011, so super recently. But he's already become a world-leading expert in blue carbon and marine paleoecology. His CV is one of those that you look at it and you go, wow, it's like more than 4,000 citations already, crazy publication record, and he is a really lovely man. So without further ado, over to you, Oscar. I'm really looking forward to this talk. Thank you.
Hi, everyone, and thank you very much, Adriana, for this lovely introduction, and Aaron as well for inviting me to give a presentation. Today, my idea is just to give you a snapshot of the research I've been doing mainly in Australia over the past 9-10 years. And I try to put quite a lot of pictures and highlight a few curiosities as well to grab your attention. And if you have any question, feel free to interrupt me at any time, or otherwise at the end, we can have a session of questions. So the title of my talk, Blue Carbon, Paleo, Shipwrecks, Policy, and plenty more to discover. First of all, I would like to start with a curiosity or a weird thing that used to happen in Australia. So this is the front page of my PhD. And essentially, it tackles two research lines. One is the biogeochemical sink associated to seagrass meadows, and this includes blue carbon, but other chemical elements. And then the use of seagrass sedimentary records as archives of ecological change. And then as well, because in Australia, they don't understand that the Spanish people can have two surnames. We typically keep the mother and the father. So they put together, and if you take my two surnames together and delete a few words, then you end up with seagrass, which is kind of a funny thing that a friend made me realize. And sometimes I wonder whether this is destiny or something. And then moving forward, this is a beautiful picture of a seagrass meadow in Posidonia in the Mediterranean Sea, taken by a good friend of Adriana and I, Quique Ballesteros, and it kind of showcase that they can be beautiful. And it's not only about what you can see, it's also what they hold underneath, that I will show you soon, some of the details and the magic that's stored underneath these meadows. And we realize over the years that they are biogeochemical sinks of pollutants and everything, you name it, even microplastics. It's been shown recently. And then there are time capsules of the past. And why? So the seagrass meadows are located in coastal areas, typically. And they receive inputs from terrestrial runoff and also from the ocean, from the plankton and suspended particles. And the canopy acts as a trap of particles. And then what happens is that the plant needs to react growing vertically to avoid burial and asphyxia. And also because the conditions are underwater, there is no oxygen in the sediments, in the substrate, and the organic matter gets preserved. And therefore it forms this organic rich sedimentary deposits that they grow pretty fast as well. And also, if you add on top that seagrasses are ranked amongst the most productive primary producers on Earth, so it provides these unique conditions. Also, because of the canopy, the sediment is protected from erosion compared to bare sand, and altogether brings this stability and the formation and modification of the substrate. The picture you can see here is what we call a seagrass escarpment or seagrass reef. It's formed because of erosional processes, typically waves or currents, and it just exposes the organic rich deposits underneath the meadow. This is another picture from the Mediterranean Sea as well, from the Balearic Islands. And this is a panoramic, and you can see the green canopy on top, and then the reef escarpment at the edge. This one was about 3 meters high and 1,200 years in age. The biggest or the highest seagrass escarpment reported to date, it's about 40 meters of exposed escarpment, and this is in the Adriatic Sea. But maybe there is even bigger, or that one in the Adriatic Sea goes further below what you can see. So imagine that you're diving and suddenly you spot like a 14-meter wall in front of you. So this is probably equivalent to 6 to 7 levels in a building. And then for what we use these sedimentary records. So we studied the biogeochemical sink, carbons, nutrients, pollutants, even sediments, and to protect coastal erosion, plastics, etc. I will show you some results today. And also we can use it to reconstruct environmental change over decades, centuries, or millennia. Seagrasses have been present in the current coastlines, at least over the last 6,000 years. This is because about 10,000 years ago, with the interglacial periods and the melting of the ice in the polar regions, the sea level rise. But then over the last 6 to 4,000 years, it's been stable. So we estimate that seagrasses established in the current shoreline about 4,000 to 6,000 years ago. And then when we look at the chronology of these sedimentary deposits, we found that the creation, so the rates of formations of these deposits are quite variable, but more or less it's 1 or 0.1 to 0.4 centimeters a year. So it's very little. It's just a very, very tiny amount a year. So they take very long to form. But then also this information tells us that if we collect a sediment sample, a vertical sediment sample in these seagrass deposits, and we slice it at about 1 centimeter thick along the core, then every single slice will have a temporal resolution of 2 to 10 years. So this means that every sample that we analyze will encompass a story of about 5 years or 2 to 10 years. Right, so after this quick introduction, I structure my presentation in 2 topics. One is the blue carbon. That's been one of the main research I've been conducting here in Australia with my colleagues. And the other is the reconstruction of environmental change. And to finalize, I added some curiosities or some research spin-off based on naturalistic observations mainly. Blue carbon. First, I will talk about the drivers. So what makes an habitat or an ecosystem more effective in sequestering carbon and CO2 than others? And then which are the threats that these blue carbon ecosystems are facing? And then highlight that we started providing estimates at regional scales, but then we also moved to global scales. And finally, we've been in contact with the government, and we are trying to move or to inform policy forward. Blue carbon. In 1980s, Smith realized that the macrophytes, they actually cycle carbon and they can constitute sinks and therefore be very important in the ocean's carbon cycle. And over the last decade, there's been a big push, a big research movement to estimate the value of these coastal vegetated ecosystems, namely seagrass, mangroves, tidal marshes, kelps, and even the Melaleuca and Casuarina forest that they are located in the upper tidal range. And essentially, it's the role of these ecosystems in capturing greenhouse gases, or actually carbon dioxide, and therefore contributing to mitigate climate change. One of the first studies we did over here is just to assess how blue carbon changes along a depth gradient. You probably know that seagrasses are limited by irradiance, and the depth limit distribution of the seagrass, it's typically linked to the minimum light requirements for the plant. And because of the decreasing light with depth, with water depth, the meadow structure adapts to it. This means typically that you have a decrease in seagrass productivity, shoot density, cover with depth. And as you can see probably in this image. And then we found that the meadows located at 2 to 4 meters, they are more effective in sequestering carbon than those located towards the deeper limit of distribution at 6 to 8 meters. And then also we realized that seagrasses store more carbon than bare sediments. And this actually makes the first important point. So if you have seagrass, carbon sequestration is enhanced. Then we move forward a bit with more the biogeochemistry, right? So why? Why some meadows have more carbon than others? In soil science, the amount of silt and clay or mud is a big driver of carbon storage. Why? Because fine particles have a high capacity to absorb carbon, so to actually bind carbon on top of the particles. And then also they are conductive to anoxic conditions, so they limit the permeability of oxygen into the soil. And all these are well-known factors that are conductive of organic matter preservation in soils. So then we put all the data together that we had, and first we started exploring trends between bare sediments and organic carbon content. And we found quite a significant trend explaining about 78% of the variability. So guys, don't look at the right graphs, look only at the left graphs. And then when we look at the very small seagrass species like Allodule, Allophila, or Zostera, so we found that this trend remains. And this somehow told us that the contribution of seagrass matter to the soils for this short living species was lower. So therefore, the impact of the seagrass on the soil composition, in this case, the mud content, but also the origin of the carbon was little, and the regression between mud content and organic carbon content prevailed. However, when we look at larger seagrass, such as Amphibolis and Posidonia, we saw that this expected relationship was lost. And then also when we look at the origin of carbon based on the stable carbon isotopes of the soil organic matter, we realized that there was a positive trend. So meaning that when you have a large seagrass that is highly productive, and then part of this biomass that scores ends up preserved in the soils, then the expected relationship between mud and organic carbon content was lost, because the majority of the carbon present in these meadows was not actually bind to the sediment particles, was actually coarse material from the seagrass plants itself. And then we created this kind of nice graph, just highlighting a bit which are the drivers or the main factors influencing carbon storage in seagrass meadows. So it ranges from biological, geological, and hydrological and chemical factors. So it's a mix of factors with their interactions, and so it's a complex thing to understand. And then one of the spin-offs as well, more linked to the chemistry of the plant biomass, the seagrass biomass, is that we collaborated with a team in Spain that they are experts in lignins, in plant lignins. So without entering details, they characterize the chemical composition of the lignin and structure of seagrass Posidonia australis in Australia and Posidonia oceanica in the Mediterranean. What they found is that the lignin of Posidonia, in particular the oceanica, is unique among all plants on Earth, has a unique structure and composition of the lignin. And this is mainly driven by the presence of phenolic compounds, the p-hydroxybenzolate. And this in more common trends, it's a paraben. So it's a phenol compound, typically named as paraben. Paraben are used as preservatives in cosmetics and pharmaceutical and even food. And therefore we realized that this could be what they call sustainable or a friendly, environmentally friendly or natural paraben that could be applied in industry. And then just very general as well, the plants, the seagrass produce these phenolic compounds as a defense mechanism for herbivory. And probably Adriana knows much more about this than I do. Right, and then also because this lignin is unique, so it's actually very hard to decompose, and that's one of the reasons why seagrasses are very effective in sequestering carbon, because the biomass is very hard to decompose. It's not attractive to microbes at all. So then there is the possibility to expand the gene responsible for the production of this PBA into trees, for instance, populus, that they are used for harvesting then for the production of paper or furniture, etc. So there is the main message from this slide is, look, we don't know very little about seagrass in terms of applications, plenty of applications for industry, pharmaceutical, medical applications from corals and sponges are being pursued, but not for seagrass. So just to show that seagrass, they might have some hidden secrets as well. Then in terms of threats, so seagrass have been decimated in Australia and elsewhere over the last 30, 40 years. Typically, it's direct disturbances, moorings, coastal development, port infrastructure, fishing practices, etc., but also because of indirect disturbances, mainly eutrophication. So an increase in the fluxes of nutrients that cause algal blooms and the die-off of the seagrass. And more recently, due to climate change and in particular, the increased frequency and intensity of marine heat waves. So just to illustrate, because this also has implications for the implementation of blue carbon into policy, if we want actually to manage blue carbon ecosystems to reduce our carbon footprint and mitigate emissions. So in here, we simply went to Rottnest Island, that it's a beautiful island here in front of Perth. And we collect cores in seagrass meadows and then in the adjacent scars from moorings. And in this graph, you can see that meadows, the seagrasses have more carbon.
I'm not sure how long they plan on going for.
So there is someone with the speaker off. Is all good? Any questions? I keep going. So another threat that we studied is the marine heat waves. 2010-11, there was a big marine heat wave along the WA coast that killed around 1,000 square kilometers of seagrass meadows in the World Heritage Site of Shark Bay. So DVCA, the Department of Biodiversity and Conservation, have been monitoring the extent of the meadows, and they provided the maps. And then we essentially estimated the amount of carbon that potentially was released after the die-off of the seagrass. And this again, it's informing or providing relevant information to inform policy and management. Then, due to eutrophication in Cockburn Sound, that is one of the most industrialized embayments in WA, there was a massive loss of seagrass since 1980s. I mean, massive, very small relative to Shark Bay. It was only 30 square kilometers. And we did the same. We collect cores on seagrass and bare sediments and then compare the stocks to estimate the change in soil or the loss of soil carbon. One of the interesting findings of this study is that if you lose seagrass, not always will entail a loss of carbon. And this is mainly because you need a high hydrodynamic energy, estimated as a shear velocity here in meters per second, to actually cause the resuspension and erosion of the soil once you lose the protective effect of the canopy. So we estimated that a threshold of 0.2 meters per second was the trigger of erosion of soil carbon. And then just very briefly, we've been coring, let's say, we've been hammering pipes all along Australia and in some places around the world. It's very fun because you get to interact with locals and then also to be in the field. That I think it's one of the beauties of being a researcher in marine ecology. And in Australia, so very quickly, some of the global estimates, that was a seminal paper by Furkurian that actually put seagrass in the blue carbon map. And it showcased that compared to other main primary producer ecosystems in the world, seagrass were well positioned, so they play a key role as well. And then in terms of specific to mangrove, we did a study to estimate blue carbon in mangroves along the world, and then based on the pressures in each country, we estimated which countries can benefit more from the protection of these blue carbon ecosystems, and also which are the countries that they are at more risk of emissions from the deterioration of these blue carbon of these mangroves. So that was interesting, and again tries to send a message to policymakers, because at the end of the day, I believe that science should inform policy and not vice versa. So we try to provide solid scientific evidence to then support like change or conservation or restoration of policy to promote that. And then also there is the white carbon, let's call it this way. So blue carbon, we always talk about the organic matter, the organic carbon, but these blue carbon ecosystems also support calcifier organisms. So they support these organisms that live and thrive because of the existence of the habitat. And this is particularly important in seagrass meadows, that they actually support plenty of gastrops, mollusks, etc. So, and the point here is that the process of calcification actually emits CO2. It's a net source of CO2, and therefore somehow we need to remove the effect of carbon or subtract the emissions of carbonate calcification from blue carbon estimates. And then this was the manuscript that actually triggered policy in Australia. So we estimated blue carbon worldwide in Australia, Australia-wide, for the three main ecosystems: tidal marsh, mangroves, and seagrass. We realized that Australia is a hotspot. It has about 11% of the global blue carbon resources. And we produce also this report for the Australian government to tell them, look, there is an opportunity here to offset Australian emissions. And currently the Australian government is developing a blue carbon method for the inclusion of blue carbon projects in carbon abatement schemes, and even the emissions reduction fund in Australia that aims to provide economic incentives to offset emissions or to capture carbon. And then also we collaborated with a few institutes in Australia and also around the world, just trying to push the science and the policy forward in these countries. Right, so next topic is the paleoecology. The deal with seagrass is that they hold information from terrestrial and marine ecosystems. And then also the paleo allows you to bridge between nature and humans, past and present. So it's a library, and then you can become storyteller somehow. First, I would like to emphasize that the Earth, as we see it today, it's not the same as used to be, maybe except few very little regions in the world. And then it's very important to have this long-term perspective of change to understand the thresholds of change and also to inform management how. So if ecosystem change, what's the ecosystem that you want? And based on what? Based on the ecosystem services they provide, based on their beauty and on tourism, what it is. So what actually do you want? And then the fact is that the human population is exerting like a pressure of about 40 gorillas, 1 ton each per square kilometer. So it's essentially not sustainable unless we do things very well. And then why is important to the temporal scale? Because essentially, once you look at an ecosystem for a monitoring program of 10 years or at present just one visit, you see only a part of the story. You see a temporal window. And the ecosystems, they are not stable, they shift, but they are resilient. So this graph, the semicircles illustrate an ecosystem status, and then the movement of the ball, it's the resilience, so the capacity to cope with change. And ecosystems change with time, and depending on the time scale that you are assessing the condition, you will see one thing or another. For instance, I'm putting some areas of the Great Barrier Reef as an example. A few decades ago, coral reefs were beautiful. Then they start degrading because of global warming and other pressures. And then in some areas, the corals are lost and a macroalgae ecosystem or a green algae ecosystem is now in place. So it changed. Monitoring allows you to reconstruct from the onset of monitoring until you finish. But then the paleo, the reconstruction, allows you to go back in time. And this is kind of the data that you can obtain from a sediment core or from a sediment register or other environmental archive that could be ice, stalagmites, lake records, or peat box. So there are several natural archives on Earth that has been used to reconstruct the past. And this data is real from Australia, and it shows that over the last 3 millennia, the conditions have been more or less normal. There's been some natural cycles and shifts, and this we call kind of the background conditions. But then over the last 200 years, there's been a big shift in the ecosystem condition. So allows us to determine what was the past condition and what's the present trend or present condition. And also for management, it's very important to identify the thresholds. Right, and to illustrate this theory, I would like to show you a couple of case studies in Australia. One is at Oyster Harbour in southwest Western Australia. That's one of the first settlements by Europeans in Australia. Coastal development occurred all over the place, and this had severe impacts in the dynamics of the wetland at Oyster Harbour. So essentially, in the 80s, about 80% of the seagrass cover was lost because of eutrophication and algal blooms. And what we did here is collect a sediment core in seagrass. Then we date the core using radioisotopes and realized that the core was about 30 centimeters long and 600 years of age. and then identified a bunch of biogeochemical proxies or variables along the core. And then using statistics, sequential regime shift to detect changes in the trends of the variables, we spotted two main periods. One corresponding to 1850s, essentially the onset of land clearance and coastal establishment in the region, and another one after the 1960s that actually was the loss of seagrass. The horizontal red lines illustrate the shifts. The first one is for carbonates. As I mentioned before, seagrass, they support calcifiers. And this indicates a loss of ecosystem structure. So the meadow was less capable of sustaining these calcifiers. And this indicates early stages of the deterioration of the seagrass ecosystem functioning. Then when we look at the sediment grain size, this is matte and very fine sands, we see an increase also over this period. This is mainly due to the land clearance and the associated runoff from soil erosion. And then when we looked at the stable carbon isotopes, we also spotted a shift. This proxy essentially tells us the origin of organic matter accumulated in the record. And it essentially shows a shift from mainly Posidonia matter to an increasing contribution of algae and other plankton and primary producers. And then the trigger here was the phosphorus. Phosphorus is a limiting nutrient in the oceans, and we spotted that when that nutrient or phosphorus concentrations were above a certain threshold, the seagrasses were lost. So the temporal match was pretty good. And I will not describe the ones in the bottom, but I will make the point that these thresholds of ecosystem change can inform or can provide target levels for environmental managers. For instance, imagine that you're an environmental manager and you monitor the stable carbon isotope composition of your surface sediments. So I would say that if you go below -10 per mil, you are in trouble. Your ecosystem, your seagrass meadow could tip towards an unhealthy state. And similarly, if your phosphorus concentrations in the soil are above 150 ppms, keep an eye, act soon, or because your ecosystem is close to collapse. So this is the kind of information that we aim to provide, but it's at the onset. We have to develop further studies. Right, so the second case study I want to show today is in Port Pirie. Port Pirie is in Australia, is near Adelaide in the Gulfs, in the South Australian Gulfs. And it's a very fucked up location. Why? Because they have a smelter operating in the area for more than 100 years. The smelter is providing the livelihoods to the settlement in the area. So most people living in Port Pirie actually work or benefit somehow from the smelter. But the smelter is creating severe health problems. So a smelter, essentially, they collect ores from Australia and then they melt them to produce metal. So that's what the smelters do. And this entails pollution. So this is to the point that the lead levels in the population, and in particular children, the lead levels in the blood of these people inhabiting the area are above the healthy levels. And then you cannot eat seafood because then you can get arsenic or mercury poisoning. So it's actually pretty bad. And it's a kind of double-edged situation. You cannot just shut down the smelter. It's providing the livelihood. So you should shut down the whole city. So the Environmental Protection Agency in South Australia contact us to try to demonstrate whether the smelter is the pollutant in the area, whether the measures implemented by the smelter to reduce pollution were effective. And that's what we did. We collect sediment cores in the seagrass nearby the area, Posidonia australis in this case, showing you the main elements, lead, cadmium, and zinc, that they are emitted by the smelter. And there was a massive increase in their concentrations following the onset of smelting operations around 1890s. So this made a first point. So yes, concentrations increased enormously since the onset. But then that's not enough. One could argue that this area is naturally rich in pollutants, and the Industrial Revolution is actually the cause of that increase. So then we analyze another proxy named lead isotopes to fingerprint the origin of the contamination. So essentially, the lead isotopes tell us the origin of the ores. So every single ore or rock on Earth has a particular ratio of lead isotopes. Then if we plot the ratios, like in this graph, we plot then the values that we detected in our core and also the lead ratios of the ores emitted or of the main ores used in smelting operations in Australia, then we get this pie plot. So the deal here is that this circle, red circle, shows the background lead isotopes concentrations from 1,000 to 3,000 years ago. Then, with the onset of smelting operations, the isotopic composition shift towards this yellow, green, and blue. And this actually is the signature of the ores imported by the smelter for their operations. So actually, this is targeting that ores from 5,000 kilometers away are in the area. So this is targeting the smelter as the cause of the pollution. I will skip this one and then start talking about some curiosities. So seagrass, because they form these deposits and they cover things. This means that they cover archaeological heritage as well. They cover shipwrecks and settlements and archaeological artifacts, and they cover it and they seal it as well. So they protect it from deterioration, from hydrodynamic energy and oxygen and microbes. So they're actually beyond the time capsules, that it's the paleo topic. They are actually security valves of ecological heritage. And to illustrate this, this is a graph of the ancient settlements in Denmark or archaeological sites in Denmark. And then if you match the graphs of archaeological sites and seagrass distribution in green, then there is a massive overlap. So first, humans like to settle in protected environments as seagrasses like to thrive as well. So there is an overlap in terms of habitat preference, but also then they discover plenty of archaeological sites underneath the current meadows. Then also more curiosities in your right, there is a shipwreck. As you can see, the distribution of this, where it's called the ceramic pieces, it's pristine. So it's actually how it's seen. And this is because the meadow was protecting the archaeological setting from erosion. And then the other picture in the left shows how here in Perth, in Corcoran Sound, they are trying to mimic the effect of seagrass by the use of these plastic stripes to preserve, to bury again this shipwreck that was studied by archaeologists. So because the seagrass was not present anymore in the area, and therefore the seagrass was not capturing and generating these sediments and burying stuff, then they try to minimize the effect of seagrass by using these plastic strips with no luck or very little success, I would say. And then this is just a very quick reflection that's important to keep your inner naturalist alive. And then I learned a lot from Quique Ballesteros, I think is my model, is escaping to the field as much as he can. And I think that's the way to really enjoy, because the reality is that when at least I became a marine biologist is that because of the beauty of the nature, not because of the beauty of my laptop. Actually, it's not that good. And the reality sometimes actually is that we dive in shitty places, so degraded places. At least myself, I don't often see reef and fish. Mainly it's in very silky and murky coastal estuaries, etc. And then we spend lots of hours in the laptop. So I encourage everyone to go in the field as much as they can. And then these are some of the curiosities we just observed and we end up publishing as well. So if you look at the structure of these two seagrass escarpments, so one, you can see some differences. One is Australis in Australia and the other Oceanic in the Mediterranean. And the Mediterranean sister is much more effective in capturing carbon. But then when you look at this, what's the most striking thing that you see? So it's actually the growth of the rhizomes. In one it's vertical and the other it's horizontal. So that's the trick. So that's why one is more effective in sequestering carbon, because it accumulates faster. And then I will quickly show you this video of another curiosity that we observed while diving in Shark Bay. So you see all these groupers. These guys actually excavated caves in the escarpments, and the caves hold because of the fibers present and the very muddy composition. So they are hiding there, and they created this habitat. So this is a role of the escarpments as 3D habitat for reef fish. And that's another reef species, a pogon. And all this hanging is the seagrass fibers. And we spotted the same in the Mediterranean, but unfortunately it's quite overfished, and fish were more little, but still is another pogon species, and you can see these caves. And even this is a rock fish that just actually has a beautiful camouflage with the colors of the escarpment. So this just was a curiosity, and then we published a couple of manuscripts. One in Frontiers in Ecology of Environment that has had the section of this, just a few words. If you discover something that it's novel and it's nice, a nice story, you could target that journal. And then that's another one with my brother, that's also a marine biologist. And he had a coral in an aquarium, and suddenly he was not taking care at all of these little poor guys in the aquariums. So they start to stress, and then they started to do the polyp bailout. So essentially they expel the polyps to try to find a better habitat for them. And that was novel for this species, was not described before, and we just published a little note based on this naturalist observation. So yeah, we have plenty of ongoing projects. Soon it will be more based in Spain than in Australia, but I still planning to keep my tides and research here in Australia somehow. And yeah, hopefully everything will be right and we will keep funding new interesting things. Thank you.
Thanks so much, Oscar. That's a really fascinating intersection of a few different topics. Would anyone like to ask some questions about those topics? You've got a number to pick from. Also, feel free to turn your cameras on at this point. I have a little crowd effect. Hi, Adriana.
I was just curious about this coral polyp thing. So can you explain that a little bit more? Like what happens with the polyps? They swap location basically.
Yeah, so the colony expels some polyps, and then I think they think that the polyp will roll in the sea bottom and maybe settle in a more suitable location. So that's a reproduction mechanism that was described for some species, but not for this one.
Sorry, and if nobody else goes, so you know this growth of Posidonia australis being more horizontal and Oceanica more vertical. I mean, why do you think that is? I mean, what's the cause and what's the effect? You know.
So yeah, certainly has to do with the habitat. I think it's mainly probably the I don't really know, but my guess is that the dispersion mechanisms. So I believe that once the Posidonia oceanica is established, then it cannot grow because it's very dense. It cannot grow horizontal, and the only choice is to grow vertical, and then this could be driven by the depositional environment. So if you receive more sediments or you generate more carbonates, then you also would be forced to start growing vertical. And Australis, I don't know. I know that the main recovery mechanism of Posidonia sinuosa and Australis is by rhizome elongation. So they spread the rhizomes horizontal. And I don't know, I believe there is a temporal reason there. So if the Oceanica has been established there for very long time and actually cover 100% of the habitat, then only has the chance to grow vertically.
I have a question, Oscar. I really like the paleo side to things, and that sort of baseline information is kind of what we're always looking for. And I was wondering if you had any thoughts on something that might be possible on rocky reefs that wouldn't have, you know, that sediment history.
So one of the most common archives, it's corals. They collect cores in corals. But then think about it, you are very limited in the variables or proxies that you can analyze. Essentially, you have a carbonate rock where to run analysis. Seagrass is much more rich because has everything, has pollen from the terrestrial environments, has all the fauna or biocenosis inhabiting the meadow, etc. So it's very rich. I believe in coastal areas, maybe the marl located at the deep edge of the reef could be a quite stable depositional environment. But I did see a couple of studies, I think, you're looking at the spicules of sponges, etc. But beyond that, maybe, no, I don't, maybe the caves as well, if there is kind of a depositional environment in the cave. But there is not much, I would say.
Yeah, the kelp history is just lost to time. So would you say, what was at the edge? The marrow?
Yeah, it's a specific term. I don't know if there is a better term in English, but it's called marl. So it's the fragments of corals and corallinegenous algae that accumulate at the bottom of the coral escarpments or coral reef edges.
Right. But still more tropical. No?
Yes, but essentially you have to find the depositional environment.
Yeah.
So an area that does not receive big waves, currents, or tides, and then it's protected from erosion. Sediment grain size can give you a very good indication of the depositional environment, whether it's the composition itself with percentage of mud or even the sorting of the particles. So you could explore a few sites, and then the true is geomorphology. Geologists will understand better as well the evolution of the shoreline and etc.
Cool. Does anyone else have questions?
But you may find as well kelp DNA or pigments in the seagrass records. So
Right.
we published a couple of studies looking at pigments, a record of fossil pigment in the course.
Right. And I guess if they're close enough in location, you wouldn't assume that the background concentrations would be all that different over such a large time scale.
Yeah, look, one of the issues that we spotted with the pigments is the decay. Some pigments decay quite faster, but I would say that you will find pigments of kelp. And then because pigments are kind of spread across, so they are not unique identifiers of a single species, I believe you would need to run as well DNA analysis to try and get semi-quantitative data of kelp abundance that you can then link to pigments somehow.
Very cool.
I have a quick question if no one else has questions. Thanks for the talk, Oscar. It was really fascinating. I had a question about the fish creating habitat kind of in those seagrass escarpments. This might be a silly question, but is that like a problem for carbon sequestration? Like, are they kind of like digging it out sort of thing? Like, do you have any idea of what it kind of does to the actual seagrass habitat?
Thanks, Shannon. So yes, essentially it is. The way these escarpments evolve, it's like the coastal cliffs. So essentially they erode at the base and then the hanging edge collapses. So they actually evolve. And the fact that the fish are digging holes, I think they enhance the erosion. And then because the materials are exposed to oxygen, so this will decay faster. There are a few studies also looking at the role of crabs or crab burrows in pumping oxygen into this soil carbon and what they input. So yeah, but these fish escarpments, it's kind of my favorite thing at the moment. I really love the story and it's super cool.
Yeah.
And I think they are cool. I will not study the impacts on blue carbon, in other words.
Yeah, no, it's really interesting. Thank you.
Anyone else?
I had a quick question. I was wondering with the work you did making reports for policy and government, have you found that to be successful? Like your work's actually getting implemented into policy, or like maybe not as much as you'd want?
Yeah, that's a very good question. So I think the policymakers, they have their agenda and you cannot modify the agenda. Maybe you can influence it a bit. So, and they want to drive the agenda forward very quick, if that's what they want. So, but at the same time, they have the need, by law, I assume, to ask the scientific community and also the citizens. So yes, overall, I was very happy with the process. They had these meetings first with some scientific background, then engaging also all the stakeholders and having these group discussions, etc. But at the end, so that yes, they took it into account. Actually, in particular, we actually ranked all potential activities that may influence blue carbon based on accuracy of cost of implementation, potential carbon abatement, feasibility, etc. And then the activity that we ranked higher is actually the first one that they selected to run a methodology. And this was the reintroduction of tidal flow in wetlands. So because wetlands are nasty, and back in the days they bring several diseases linked to mosquitoes, etc., they were dry out. And now by just destroying a levee or a wall, you can reintroduce the flow and then enhance the recovery of mangroves and tidal marsh. So the Australian government is developing this methodology that then will allow any company, entrepreneur, etc., to sign up a project and start gaining carbon credits. And these carbon credits as well, they contribute to Australia to meet the Paris Agreement for climate change mitigation. So, and overall, at the end of the day, the beauty of all these probably is to promote the restoration and conservation of these wetlands.
Awesome, thank you.
I was also wondering, how did you become involved in that, Oscar, and creating that report?
CSIRO was commissioned a big project called the Coastal Carbon Cluster involving about 12 institutions in Australia. And that was the big effort to quantify the blue carbon in Australia. I played a big role in driving this project forward, and therefore they dragged me into this policy framework.
So were you employed with CSIRO at the time, or is it like independent contract?
Yeah, actually for that particular report, and because I was a bit loose, contractually speaking at the CU, I was employed by CSIRO for a short period of time, I think 3 months, specifically to produce this report.
And they approached you, or you responded to an advert?
So at first instance, they approached me. At this later stage, towards the development of the method, they kind of bring a community of experts that they run all the workshops and assess methods, feasibility, etc. And this was, and I believe it still is, open to everyone interested, from companies, universities, NGOs, etc.
Yeah, really interesting. On the groundwork. Do we have any last questions for Oscar?
I'm gonna have to step out for another meeting, but thank you so much. That was brilliant. Thank you, Oscar.
Thanks, Adriana.
Don't forget the barbecue. I guess I can segue that into everyone. Don't forget the barbecue. It's in half an hour on the lawn downstairs. Oscar, I guess your burger will be in the mail or in a t-shirt or some other format. But thank you very much for joining us. Everyone else, if you have any ideas for speakers, you can shoot me an email and we'll continue to organize this seminar series throughout the semester, year, however we go. And we'll hopefully see you all soon. And thanks again, Oscar.
Yeah, thanks everyone. Very nice to talk to you.
Thank you.
Bye.
Thank you.
See you.